Okay, good morning. Let's go ahead and start with our lecture for today. So just a quick reminder about this week. In recitation, you will have your first real quiz of the semester. So you've had an opportunity to take a practice quiz with your TI in recitation last week so that you could see how it works with having Zoom open so that your TI can see you and help proctor you during the quiz. And then you have Laulima open and you go ahead and you take the quiz in Laulima. So hopefully that went well for you. This week will be the first real quiz, and the quiz material will cover mainly the first couple lectures. You know, the first lecture of the semester was just sort of like an introduction lecture. Lecture 2, though, is when we started talking about the chemistry of the cell. And it does turn out that there are a couple questions from the first part of lecture 3, which was last Friday's lecture, where we introduced the topic of polymers and how polymers form as these very important macromolecules. So in the PDF version of today's lecture slides, I think it says the first quiz is only going to cover lectures 1 and 2, but I think that was a little outdated. So if you look at this slide on the screen here, really I want you to keep in mind that it's mainly on lecture 2 stuff and a little bit of lecture 3 about the polymers. So it does not talk about anything about proteins and things like that. That will be on the next quiz for next week. Okay, so I've asked your TIs to make an announcement and let you know that you should also take a look at the first part of lecture 3, where we talk about polymerization. In addition, you have your first homework assignment due this week. I think most of you have already at least opened it up and taken a look at it. Keep in mind that pretty much every week you have a homework assignment that's due by the time your recitation rolls around again. You're allowed to work together on the homeworks. The way I see it is that learning oftentimes works a lot better if you have classmates. These days you can do it over Zoom, right? And you can kind of bounce ideas off of each other. And I always felt that I learned a lot better if I had classmates to kind of work with and study together with. So absolutely, you are allowed to work on your homeworks together. However, you are responsible for knowing what it is that you are submitting, right? Because if you're just copying each other's work and then submitting it to get points, you're going to be in trouble when it's time for the exam and you're on your own doing the exam. All right, so make sure you understand the answers that you're putting down on your homeworks, even if it came from working together with a fellow classmate. Okay, good. Let's quickly review so that we're back on the same wavelength here about what we were introducing in this class so far. We said that there are 4 main macromolecule families that make up the components of our living cells, and therefore all life on Earth, because it's based upon cells, you could say that these are the 4 main macromolecule groups that make up all life. Right? So these are things like polysaccharides, proteins, nucleic acids, and lipids. We said that 3 out of 4 of these groups are truly what we can consider polymers, meaning that they are long molecules, large molecules made up of repeated units of smaller molecules, whether they are simple sugars like a 6-sided glucose here, that's a monosaccharide being chained together to form a polysaccharide, or an amino acid, right, which we looked at on Friday, that all have the same standard structure of a carbon with an amine group, a carboxyl group, a hydrogen off of it, and some sort of variable other functional group here, which we often refer to as an R group, right? And we said that life on Earth uses 20 different variations of amino acids, and they differ just by these R groups over here. Amino acids can be strung together to form proteins that then fold up and can be structurally important or important as little machines. Nucleotides that are made up of 3 components, which we will study later in more detail, a ring sugar with a nitrogenous base and a phosphate group, that itself is sort of a basic unit, a fundamental unit, a monomer that can then be linked together to form a much bigger polymer, in this case, a nucleic acid. We said that the fourth group of macromolecules, lipids, are just as important as the other three, right? But they're not truly polymers. Why? Because lipids are really just long chains of repeated hydrocarbons. So carbons with hydrogens on them. And as a result, there really is no discrete single unit that you could say, oh, this is a fundamental building block of a lipid. It's just a big chain. So I said it's like a big braided rope, right? If you have a big braided rope, you don't really see discrete units that make up the rope. Unlike something like a chain link, that each link you can very clearly tell is its own unit. So we will spend the next few lectures going over these in each more detail, these different families. So today, let's start off with polysaccharides, okay? Actually, I should say we already talked about proteins, an introduction to proteins last Friday, right? So today we're moving on to polysaccharides. Polysaccharides are sugars, right? Sugar is the common everyday name that we give to these things. The typical person on the street, if you say a, polysaccharide, they might not know what you're talking about, but you say sugar, yeah, okay, they know what a sugar is. A sugar technically is what we call a hydrate of carbon. Right? Now that's different than hydrocarbon. Remember, last time we looked at some examples of hydrocarbons, things like methane or acetylene, right? Or even, I told you, like the long chains of hydrocarbons that make up something like a lipid, right? Those are just carbons and hydrogens, right? That's it. So we call them hydrocarbons. Hydrates of carbon is a little bit more complex. When you say hydrate, just think about the word hydrated, right? You're talking about not just hydrogen, but actual water molecules being involved, right? So sugars are quite simply carbons that have waters attached to them. Maybe you never really thought about it that way, but that's what they are. Right? Sugars are made by plants via photosynthesis. So this cartoon on the right here, I think you all know what this is showing, right? A typical green leafy plant gets energy from the sun, and through this process of photosynthesis that we will learn about later, right, it combines carbon from the atmosphere that's coming from carbon dioxide with water, right, from the atmosphere or water that's coming up through the roots, and it recombines those atoms to form sugar molecules. Right? So a typical sugar chemical structure is simply, for every carbon molecule, N, there is a water molecule, N, right? And that comes from the CO2 and the H2O, and you're going to have leftover oxygen, which the plant does not use, and it releases. So a simple sugar like this might be termed a monosaccharide. And they typically have the OSE suffix at the end, like glucose or lactose, maltose. So glucose, because it's made of 6 carbons, is what we call a hexose. You can see here are 2 ways of representing the glucose molecule. So glucose is C6H12O6. So again, for every 6 carbons here, there are 6 molecules of H2O. Here you can see it as a linear chain, right, where you have the 6 carbons, carbon 1 through carbon 6. But typically, the way that we think about glucose is not in its linear form, because this oxygen group over here actually very happily reacts with this carbon at C1. So you actually get a bond where this oxygen is kind of bridging carbon 5 with carbon 1 over here. So it forms a ring, right? And so this is usually how we view glucose. So you can see here, here's carbon 1 over here on the right, 2, 3, 4, 5, 6 is up here. And you can see this is the oxygen that is bridging the C1 with the C5, right? So it might not be totally obvious, but the ring that forms is not actually carbons all the way around, right? Part of the ring is actually the oxygen that's kind of making this thing bend around. And we'll elaborate on this a little bit more in today's lecture, thinking about things as rings. Again, this is just a convenient way of looking at a stick figure to simplify things. But hopefully in this class, as we move on throughout the semester, you'll appreciate to visualize things in more three-dimensional space. So there is an alpha form of glucose. Alpha means the hydroxyl group that's attached to C1 in this drawing is pointing down. Right? Now, remember, because it's three dimensions, the hydroxyl groups don't necessarily, right? They're not all flat like on a piece of paper. They are actually sticking up and down from this ring. Right? So this C1 position hydroxyl group can also point up, and it still would be sterically favorable, in which case we would call this a beta glucose. Right? I know you've been through this type of thing before in OChem, but I remember when I was taking OChem, I always thought, well, this is just sort of like you're naming every single position of one thing being in one position, one thing being the other. Why does it matter? Right? Because it's still the same C6H12O6. It matters again because the shape of the molecule is slightly different if that hydroxyl group at C1 is up versus down. Okay? And that matters because in all of molecular cell biology, right, and biochemistry in general, the interaction of molecules with other molecules is dependent upon those molecules being the exact right shape to interact. So something like the hydroxyl group pointing up or down can make all the difference in the world as to whether or not some other molecule can properly interact with this glucose. So let's look at this here, slide 6, different views of a glucose molecule. So again, the simplest way we often think of things is just by stick figures, line drawings, like you see on the left here, right? This is what we looked at on the previous slide. But let's get a little bit more realistic here, right? Because if you guys are going to successfully be able to follow a lot of the concepts about macromolecules interacting together, we have to think beyond just the stick figures, right? So we can make it a little bit more complicated. Here you go. This is still a stick figure, but now instead of just letters, you actually have little balls here representing the different atoms, right? So this gets a little bit more realistic. We can go a step further and actually make it more three-dimensional. So this would be like your little OChem Tinker Toy sets that you can put together molecules, right? Now you can really appreciate the idea of three-dimensional, certain hydroxyl groups pointing one way versus the other. But this ball model, ball and stick model, still cannot really help you appreciate the overall three-dimensional shape of what a glucose molecule would look like, what space it would actually occupy in real life. So in order to do that, we get rid of these artificial little sticks, right? Because the sticks, again, I mean, what are they, right? They're meant to show that, oh, there's electrons bonded together across there. But it's not going to be really sticks, right? Because again, the electrons are in their orbital clouds, and it just shows that electrons are sharing an orbital cloud. So let's get rid of the sticks, and you get this type of a figure here, right? So in other words, this rightmost model is the most accurate way of visualizing what a molecule of glucose really looks like. And you can see here that once you really try to visualize it in 3D, the whole concept of a ring sort of becomes kind of pointless, right? I don't see a ring in this at all, right? And yet this is the real shape of the glucose. So the take-home message from this is we rely on line drawings to learn chemical reactions and maybe help us, draw things out a little better, right? Like in OChem, when you're doing different types of nucleophilic attack and you draw arrows and stuff like that. But I want you to never forget that a molecule's true three-dimensional shape is what's going to determine what kind of reactions can occur between this molecule and another molecule, right? And more importantly, how they occur, what surfaces of this molecule are actually the right shape to fit with other molecules. So the notion of shape is especially important in molecular cell biology when we start talking about how different types of macromolecules work together, how they come together to achieve amazing things on the molecular scale. So to continue with our discussion of sugars, simple sugars, simple monosaccharides like glucose, like galactose or fructose can come together to form disaccharides. So this is kind of like the start of thinking about polymers, right? So the simplest kind of polymer would just be like a disaccharide. In this case, two glucose molecules, two alpha glucose molecules. We know they are alpha because you can see the OH group at the carbon 1 is pointing down. I mean, you don't see the OH labeled here. On this one, you can see the OH is pointing down, right? So both of these are alpha glucoses. And they form a bond together between the C1 position, the carbon 1 of this over here, and the carbon 5, sorry. That's bad. 1, 2, 3. No, carbon 4. Sorry, that's really bad. 1, 2, 3, 4, right? C1 to C4, right? And this is what we call an alpha glycosidic bond. Glycosidic bond means a bond that's formed between two sugar molecules, two sugar monomers. I put a little asterisk here to remind us that this happens via a condensation reaction, right? So in the formation of this bond, a molecule of H2O is released. So two glucose molecules like this is what maltose is. So maltose is a disaccharide. And maltose sugars come from things like sprouted grains. And this is the sugar that's used to ferment grains into different types of, for example, beer, right? If we have a glucose molecule, in this case, a beta glucose molecule, because you can see the OH is pointing up here, and it's forming a beta glycosidic bond with a galactose. So that's what this is over here, right? So notice this bond shape is different than this bond shape up here. This is what lactose is. So lactose, that's found in milk, right? So lactose then is a disaccharide that needs to be broken down in order to get things like the glucose out of it so that your body can use the glucose for energy. All mammalian milk has lactose in it. I think you're aware that some people, especially as adults, have trouble digesting lactose, right? And so that's why you need to take dietary supplements like Lactaid, right? So Lactaid is a lactase enzyme. It is a protein that comes, and that would be the protein that's able to break this beta glycosidic bond, right? Releasing the glucose from it. So again, you have to have a particular enzyme that would be the right shape to be able to work on this beta glycosidic bond in order to break it to get the glucose out of it. What happens if you don't have this enzyme? Well, your body doesn't break down the lactose, and this lactose passes down into your intestines undigested, right? Still as a disaccharide, and it ends up in your colon. And in your colon, you have a ton of friendly bacteria. That's just part of our dietary tract, probiotics, that kind of stuff. Whoa, they can digest the lactose, right? And they start breaking down the lactose. And in their process of breaking it down, they release a lot of metabolic waste products, including things like gases and things like that. And that's why you get uncomfortable and bloated, right? So people who are lactose intolerant can take the lactase enzyme to make up for the fact that they don't make it themselves. But here's the thing, right? It's not that the human body doesn't know how to make lactase, the enzyme that can break this down. In fact, we're all born with the ability to break down lactose sugars. Why is that important? Well, think about this. Cow's milk, which is typically what you think of when people are lactose intolerant and they have to take Lactaid in order to enjoy milk or ice cream or pizza, cheese, stuff like that, right? Cow's milk has about 4 to 5% lactose sugar in it, right? So you might say, well, then human milk, must not have lactose, right? Because after all, we're meant to drink human milk as a child, as an infant nursing, right? No, in fact, human milk has more lactose in it than cow milk. Human milk, breast milk, typically has between 6 and 7% lactose in it, right? So that means everyone who's born can digest lactose. So what happens? What happens is that evolutionarily, right, we are designed after we are weaned from breastfeeding, at age 1 or so, right, for the rest of our lives, we're not designed to expect milk anymore. So as a result, our bodies have evolved so that as adults, we don't bother making the lactase enzyme anymore because we just don't need it, right? I mean, picture this. A typical dairy cow, right? You imagine a cow like on a dairy farm, right, with a big udder full of milk on it. It's not like our ancestors, ancient people, would be like prowling around the savannah or something and come into a clearing and see like a dairy cow and be like, oh, there's a cow, I'm going to get some milk, right? No, that's not how we evolved. So we evolved to no longer need to make lactase as we grow up. That ability to still make some lactase varies based upon your genetic background. So some people still produce some lactase, right? But a lot of people no longer have much lactase around. And so our decision to consume dairy products, right, is just something that we've made up ourselves, right? It's not what we evolved to really do as adults. So fortunately, we can just take lactase enzyme, get it into our gut, and it'll go ahead and digest it, the lactose. All right, third example, sucrose. Sucrose is table sugar. I remember when I was a student, I used to just think that table sugar was glucose, right? Because glucose is like, you think of it's like the fundamental sugar and it's sweet, whatever, right? No, table sugar, sucrose, is actually a disaccharide. It is a combination of one molecule of alpha glucose and one molecule of beta fructose. Right? And together, that makes table sugar. The taste of sweetness, where does that come from? Right? It comes from the fact that the hydroxyl groups that are on these rings here, right, are what are detected by different taste receptor proteins on our tongue, the little taste buds or papilla of our tongue. Right? So it's a chemical reaction where the hydroxyl groups are being picked up by these receptor proteins, and then those proteins send these nerve signals to our brain, and we interpret it as sweet. What's interesting is that fructose, because of its slightly different chemical structure, actually binds those receptors, those sweet receptors on our tongue, stronger than glucose does. So that means for the same amount of each compound, glucose versus fructose, fructose is actually perceived by our brain as being much sweeter. And that's why a lot of soda companies use high fructose corn syrup as a sweetener. First of all, it's cheap because they can just get it by essentially squeezing corn, right, and processing the corn and getting the syrup out of it. And ounce per ounce, the fructose triggers a much sweeter taste in our brains than glucose does. Okay, so what I'm trying to do here is give you guys some real world examples of how the stuff that we talk about in this class actually does matter to your daily life. Okay. When you start stringing more sugar monomers together and form not just disaccharides, but long chains of polysaccharides, right? Polysaccharides are long chains of sugar molecules. This is used by living cells and our tissues for different types of functions. For example, in mammals, like this mouse or like us, if we have excess sugar available in our blood, for example, you eat a humongous meal, right? You eat a humongous meal and then you're just kind of sitting around, right, doing not much. Your body digests this, releases a lot of sugar into your blood, but you're not really using it. Your body's not going to be like, all right, forget it. Let's just get rid of the sugar because we don't need it. Because evolutionarily, hey, right, that's valuable stuff. This would be like, again, our ancestors roaming around on the savannah, right? You part the weeds or whatever, and then there you have this humongous, like, loco moco with 2 patties, gravy, 2 eggs on a big bed of fried rice, right? Okay, so you eat all that. You don't know when the next time you're going to come across, a loco moco is, right? It might be another 6 months or something. Hopefully not that long. So your body's not going to waste that energy. So any excess sugar that it derives from breaking down the carbohydrates in your meal, it can convert that into glycogen. So glycogen is when your liver cells take that extra sugar that's passing through your blood and starts just chaining them together to form kind of like a nice short-term storage molecule, right? And so you can see here, the glycogen is just chained together glucose molecules. So they're chained together again by a alpha 1,4 glycosidic bond, right? Occasionally you'll have the alpha bond formed from the 1 position and the 6 position, and you get these branches of this chain. So a glycogen molecule looks like this, very feathery, right? This big branch molecule. And I think by looking at it, you can realize, yeah, I get it. That's sort of a short-term way of storing extra glucose, because then if your body needs that glucose again, your liver can convert those long chains back into individual monomers through a hydration reaction, right, the reverse of condensation, to release individual glucose monomers back into your blood so that your cells can take it. And that's quick and easy, right? So the glycogen is your quick and easy short-term storage. Plants also do that as well. In plants, we call this starch. So here you have your nice Okinawan purple potatoes here. If you look at the starch that's in there, similar idea, right? Big chains of glucose, but the way it's branched together is a little bit different than glycogen, right? So when we eat things like a potato, we're really just mooching off of all the hard work that the plant made in doing photosynthesis to make sugars and then putting those sugars together as starch to store for another time. But then we dug up the potato and ate it, right? And now that glucose, that starch, can be digested by our bodies, and our bodies have the right enzymes to be able to break down that starch. Again, we have the right molecular components that can recognize these alpha bond shapes to be able to then do the hydrolysis reaction and break them apart to get the glucose out of it. Plants also string glucose together to form cellulose. So you can see here, cellulose is different. It is strung together not by alpha glycosidic bonds, by that slightly different shaped beta glycosidic bond. Right? So you have a beta glucose over here, right? It's being strung together with a beta glucose. As a result, plants don't do this as energy storage. They do it for structural support. So the long bundled fibers of glucose molecule, right? Those are the fibers inside of plants. And I bring this up because our bodies don't really have the right enzymes to be able to recognize the beta glycosidic bonds. So as a result, when you eat a lot of salad or plant material, vegetables, fruit, things like that, right, your bodies have a harder time breaking down the cellulose polysaccharides. Okay, it all has to do with shapes. Okay, let's move on and talk about lipids. Right? So lipids are another group of macromolecules, and they are made by glycerol and fatty acids. Fatty acids, again, really just sort of being chains of hydrocarbons. So let's take a look at this group. So lipids are long hydrocarbon chains. Okay. Let me see. Hopefully this slide moved on here. Lipids are long hydrocarbon chains. So you can see here, you have a long tail of C's and H's here. And then in this particular case, it ends with a carboxyl group on the end. Together, this is what we'd call a fatty acid, right? And a fatty acid is kind of the building, is the start of putting together different variations of long lipid type structures that are used for different purposes in our body. For example, these types of structures are used in long-term energy storage. So instead of having glucose molecules conveniently linked together in something like glycogen, and you can just break a glucose molecule off and use it, here right? You see that the energy is just stored in individual carbon-carbon bonds. So why is this better than glycogen? You can store a lot more energy per size of molecule, right? Because now you have a really tightly packed chain of carbon-carbon bonds. That's a lot more energy per size than having a bunch of glucose molecules linked together. Right? So for long-term storage, that's good. The downside of it is it's going to require more effort to retrieve the energy back out of these. It's not like you just break a glucose off and the glucose is ready to use, right? You're going to have other more elaborate ways of actually breaking these carbon bonds to harvest the energy. Okay. So long-term energy storage, also lipids are a very important component of membranes that make up the cell. Whether you're talking about the cell membrane that makes up the outer boundary of the cell or the different membranes that make up the structures inside, right? That all relies upon having lipids to make those barriers. And hydrocarbon lipids like this oftentimes float around and are used in other biological functions, for example, as molecules that are meant as messages that are sent throughout the body. So there's a diverse group of lipids, but the one thing that they have in common is that they are hydrophobic, meaning when you look at this chain of C's and H's, right, you don't see any charges on them. You don't see any polar type of atoms like oxygens that are kind of messing around with the distribution of atoms, right? It's just C's and H's, and all those C's and H's are very happy. And as a result, remember, these things are constantly surrounded by water molecules inside of our living cells. So you imagine, if this is a C and an H here, you can imagine an H2O floating around next to it, a whole bunch of H2Os floating around this thing. The water has no interest in interacting with the hydrocarbon chain. The hydrocarbon chain has no interest in reacting with the water. So all the water molecules do is they just start bumping this thing around, right? They start pushing it around just because, all the water molecules are busy bouncing around. And so these lipids get shoved around. And so we say they're hydrophobic. That's why if you have a bunch of oil and water, and you try to mix it up, right? And you let it sit there, eventually, what do you see? The oil sort of comes together again in big droplets, and it kind of comes to the surface, right? When we're growing up, we think of that as being, oh, the oil hates the water. That's why it's phobic. It's scared of the water, right? But in reality, no, what's really happening is just the water keeps shoving it around, right? All these lipid molecules. And as a result, they get shoved together, right? So you get droplets of oil simply because that's the most convenient thing that happens when they're all getting shoved around by the water, right? And they get shoved together into a droplet, and the water even wants to shove it out, right? So the oil kind of is at the top of the water, right? So there's going to be few, if any, polar side chains to these types of lipid molecules, and so they are insoluble in water. That's why oil and water separate. They don't mix. There are 6 main classes of lipids. These are fatty acids, triglycerides, phospholipids, glycolipids, steroids, and terpenes. All right, so let's take a look for the remainder of today's class of these and appreciate what the differences are. Fatty acids. Okay, so this is slide 11. Fatty acids are the building blocks of many other lipids. So as I described, it's a chain of hydrocarbons with a carboxyl group at the end. So here we have 2 examples of fatty acids. One is a saturated fatty acid, and one is an unsaturated fatty acid. I think you're already familiar with these terms from everyday life. We talk about saturated fats and unsaturated fats, right? What's the difference? In a saturated fat, every carbon along the main hydrocarbon chain, right, is happily engaged in 4 bonds, right, with different molecules. So if you just look at one of them here, this carbon here, right, it has a bond with this carbon up here, a bond with this carbon down there, and then 2 bonds with the 2 hydrogens that would be on either side of it, right? Because it has all the hydrogens it needs in order to be fully happy by having 4 bonds, we say that this molecule is saturated, right? So saturated refers to whether or not all the carbons happily have hydrogens surrounding them. If you look over here, what has happened is that they've removed a couple hydrogens, and as a result, these carbons now form a double bond with each other, so that each carbon still makes 4 bonds, but now because you took 2 of the hydrogens away, right, those bonds have to be double bonds between these carbons. As a result, the shape of the molecule ends up with a kink in it, because the bond angles here, right, formed by having a double bond and then having these 2 hydrogens sticking off the side here, causes a kink or a bend in it. So as a result, the unsaturated fat, right, is not straight. So molecules of saturated fats, because they are straight, as they are kind of, let's say, interacting with each other, right, and there's energy that's kind of moving them around, right, at a certain point, it's a lot easier for them to line up next to each other and stack up. So at a given temperature, for example, room temperature, they can stack up very neatly and they become a solid. So animal fats are a good example of saturated fats that are solid at room temperature. Unsaturated fats, because of that kink in them, at the same temperature, let's say room temperature, because of that kink, they don't stack up very neatly, and so they remain as a liquid. So different types of vegetable oils, vegetable fats are examples of unsaturated fats. So things like olive oil, right, is liquid at room temperature, whereas something like butter or bacon fat, right, is solid at room temperature. And we encounter these as parts of our everyday diets. Because these are slightly different shaped molecules, right? And again, what I want to impress upon you is it really all comes down to shape when you're talking about how different biological reactions happen inside your cell. Saturated versus unsaturated fats are utilized differently by our tissues and have different health consequences in terms of pros and cons of having saturated versus unsaturated fats. Triglycerides are 3 fatty acid tails here, right? So each of these are fatty acid. You can see there's 2 saturated ones. In this case, the third one is an unsaturated one, but this can vary, right? Combinations of saturated or unsaturated. But the point is you have 3 of them that are held together by this molecule on the left, glycerol. So glycerol is 3 carbons, right? With the hydroxyl groups on them. And so they're technically, this is no longer a simple hydrocarbon, right? Because a simple hydrocarbon would just be C's and H's, and you have oxygens in here. This is not a sugar, because if you counted up all the H's and oxygens and carbons, it wouldn't be 1 molecule of H2O for every molecule of carbon, right? So what is this? Well, it's an alcohol, right? So the alcohol is kind of in between. It is sort of like a hydrocarbon that has now hydroxyl groups, but not enough to make it really a sugar. Glycerol then acts as kind of like a clamp or a staple that can then interact with these 3 fatty acid chains in a condensation reaction. So in other words, as the OH here is interacting with the OH over here, you're going to get a molecule of H2O that's booted off by this, allowing a bond to form over here, right? So again, condensation reaction is a very typical type of reaction that happens inside of our cells to create bigger molecules from smaller ones. So when you have now 3 fatty acid chains kind of stuck together by glycerol, this is a triglyceride, all right, or triacylglycerol. And this is really what we talk about when we talk about animal fat, right? This is my expanding waistline from eating all that bacon, right? And this is how our body stores long-term energy. Again, because it's a very compact way of having lots of carbon-carbon bonds that can be broken later to get energy from that to drive other reactions, right? But because it's hydrocarbon chains, it's not as convenient as just having glycogen, where you just have glucose strung together. Fat is also useful as insulation in cold climates, right? When I was living in Boston, I could argue that I get fat over the winter to stay warm, but here in Hawaii, that argument doesn't really hold. I just get fat because I'm eating all those locomocos. It's harder to break down compared to glycogen. So other examples are phospholipids and glycolipids. Here you see we also have a glycerol, right? But instead of holding together 3 fatty acid chains, it's only holding together 2 of them. The third carbon of the glycerol has a phosphate group attached to it instead. And that phosphate group may or may not have additional groups stuck on it, R group, right? Again, remember, R is just a generic symbol, just meaning something else stuck to it that you can vary, right? This does not mean it's the same R group as the 20 amino acid R groups that you guys are learning, right? R is just a generic way of showing that there's something else there, right? So these types of phospholipids would then look something like this, right? The 2 fatty acid chains, you have the glycerol. So you notice you can't even appreciate the glycerol in here, right? Because the glycerol is these 3 carbons. You can't really even see it once you collapse this down and just think about the orbitals, but you can see the phosphate group, okay? And in this case, the R group that's attached here. All right, so what is this R group over here? This is choline, right, in this particular case, because the most common type of phospholipid that's found in, for example, our lipid bilayers of our membranes, so that's what you see here, a whole bunch of these phospholipids forming a bilayer, right, are typically the R group is choline on it. A glycolipid, same thing. It would be the same structure, but instead of a phospho group, right, the tip off here is this is going to be a glycolipid, right, meaning there's some sort of sugar group here. So I should have pointed that out up above here, right? And alcohol is very similar to a sugar, right? But it's not exactly the same formula-wise, but you can see even the name glycerol seems to suggest that it's an alcohol of sugar, right? Okay, or an alcohol version of sugar. So glycolipids have a carbohydrate group here attached instead of a phosphate group. Steroids and terpenes. So these are other examples of lipids that are very important in our living cells. You notice when you look at these orange sort of rings, you're like, wait, this isn't a long chain of hydrocarbons, is it? Yeah, it is. It's just, remember when we talked about chains of carbons being thought of as linear, drawn out, or as ring structures, like for example, glucose can be thought of as 6 carbons in a line, or you can draw it as a ring. You know, I mean, hydrocarbons, many of these fold up into ring-like structures as well, right? So in this case, it's a long chain. You can see it's numbered here. There are, 18, 19 carbons involved in this long hydrocarbon chain that then is forming these different rings, right? This 4-ringed structure that you see here is the basis of a steroid. Okay, steroids like testosterone and estrogen, so these are the sex hormones in men and women, right, are these molecules, right? So they are technically a type of lipid. Where do they come from? They come from this starting molecule here. This is cholesterol. Right? So cholesterol, again, is a hydrocarbon that is now in this typical 4-ring structure, and cholesterol not only is very important as the basis for what our body uses to make things like steroids, right? So I should say steroids are not just things like testosterone and estrogen, the sex hormones, but steroids are also things like glucocorticoids that are involved in inflammation, right? So that's why if you have a mosquito bite or a rash, you take what? Hydrocortisone cream, right? And cortisone cream is a steroid that reacts with receptors in your skin to reduce inflammation. Right? In our body also makes these types of steroids, these anti-inflammatory steroids, and they're all based upon cholesterol. Cholesterol also is a very important component of our cell membranes. So when we study the makeup of the cell membrane in more detail later in the semester, we will see that among all the phospholipid molecules that make up the bilayer are all kinds of other things that are in there in the membrane: proteins, carbohydrates, and lipids, including cholesterol. Okay. Here is on the right what we call a terpene. A terpene has this ring structure at the end and essentially a chain of hydrocarbons, right? It's a little bit more complicated because you can see each one of these, it looks like it's kind of repeating this structure here. This structure is what we call an isoprene. Now, this might seem to suggest that this is like a polymer then, right? You might say, oh, is this like polymer of isoprenes? No, not really. Okay, because again, these individual units, right, don't really have much function on their own. It's just that when you have this repeated pattern in this molecule, this is what makes up a terpene. This particular terpene is something that's actually familiar to you. This is vitamin A, right? So when you think about vitamins and you say, oh, you know, I don't get enough vitamins in my diet, or you take vitamin supplements, what are vitamins, right? What do they actually do? And you're like, well, they make you healthy. Yeah, but how do they make you healthy? They help contribute with very important enzymatic pathways in your cells that are part of cellular metabolism. And we will study later in the semester why vitamins are so important and how they actually help you promote normal cellular health, tissue health, and your overall body health. But vitamin A is an example of a terpene, right? So this is the structure of vitamin A here. Okay. So to end up with today then, I just thought I'd put this in here as kind of a fun, sort of another example of relating this to our sort of daily lives. Many of you who are maybe conscious of your diet, I mean, I keep in mind that you're all college students. I think when I was a college student, my biggest concern about my diet was, did I want pepperoni or meatball on my pizza, right? But many of you are probably aware that there are these things called trans fats that you want to avoid, and trans fats are bad, right? For a while, like in the 80s, when I was growing up, 90s, I don't think people really cared about trans fats. But then all of a sudden it became like, oh, trans fats are evil. You got to stay away from them. And now everybody, like on the label of a potato chip bag, will say, oh, we don't have trans fats. What are trans fats? Trans fats are related to saturated and unsaturated fats. If you look here, here's a natural saturated fat. So this is stearic acid that's found in butter. Again, it's saturated. All the carbons are happily occupied by hydrogens, so it's nice and straight. And as a result, at room temperature, the butter is solid. Right? Compare that to something like an unsaturated fat, like linoleic acid that comes from vegetable oil, or oils from things like avocados and nuts. Right? They are missing some hydrogens, and as a result, the carbons that are missing the hydrogens, they double bond with each other, right, to kind of make up for it, making the molecule kind of kinked. And that's why unsaturated fats are liquid at room temperature. The important thing here is these double bonds are all cis double bonds. What do I mean by cis? That means when the double bond forms, the hydrogens on these carbons are pointing in the same direction, right? Either up or down in this line drawing. So that's the natural way that unsaturated fats would look. A trans fat is if you take one of these cis double bonds and chemically alter it. So now you can see you've twisted it around. So now that the hydrogens are pointing in opposite directions. So instead of cis, they are in trans, away from each other. What does that do to the molecule? You can tell it straightens it out, makes the unsaturated fat more like a saturated fat. That's why a lot of food processing manufacturers love this process of taking vegetable oils, right, and chemically modifying them to be trans fats, because then they have a very cheap way of making saturated like fat substances. We evolved to love saturated fats, right? Saturated fats are the fats in foods that make things taste good, feel good in the mouth, right? That's why potato chips, right, that are loaded with saturated fat, that's what is that very pleasurable kind of experience eating them. So manufacturers can take cheap vegetable oils, chemically process them to make them into trans fats, right, as an easy way of then coming up with this. Unfortunately, because this is unnatural, we did not evolve to have the right types of enzymes that can recognize a trans double bond. And so therefore, These types of trans fats oftentimes don't get properly digested or tolerated by our bodies, and they can actually be very damaging to our body's tissues, right? So that's why we now know to stay away from trans fats. It might have sounded like a good idea at the time as a cheap way to make potato chips or, vegetable shortening like Crisco, right, to make your food taste better. But in reality, right, it's not what our bodies naturally expect and don't naturally know how to deal with. All right, let's go ahead and stop there today.